começar a inventar moda.
Last we saw. Question is, where's the spider? Not far enough.
We did not go straight either, but
yeah, he had.
Okay, let's talk about how to do the most exciting part about this lab, the statistical analysis of the autotroph biodiversity. So what you can see here, which should be similar to what's in your data table right now, is a list of the 20 different plants that you found at each one of the different meter markers along your transect. And I've just coded them in here with various letters. It doesn't matter what letters you use. But the second row that you see below there is where I'm indicating whether they are exotic or native. Now, one of the most important things that you have to be able to find as you do this would be the number of runs. And the runs consists of every time it changes from one species to another. So for this particular transect, I can see that there are 2 goldenrod in a row. That represents the first run. Then it switches to an arrowleaf, and that would be the second run. Then it switches back to 2 Canadian thistle. That's going to be my third run. The fourth is going to be a goldenrod. Then there are 3 purple loosestrife. That would be my 5th run. My next one would be 2 sedges. That's my 6th run. And then finally, from 12 through 20, they're all cattails. That represents the 7th run. So even though they're different lengths, the runs consist of where it changes. Once again, 2 goldenrod, that's my first run. Arrowleaf, that's my second run. Canadian thistle, that's my third run. Goldenrod 4th, purple loosestrife 5th, sedges 6th, and then the cattails are the 7th. Now what I also want to indicate is that of these 7 runs, how many of them represent exotic species? Well, we know that Canadian thistle is an exotic species, so I've indicated them right here. I also know that purple loosestrife is an exotic. So 2 out of my 7 runs represent exotics. So the next thing that we need to do is to take this data and turn it into the various indices. Now, you should have these equations somewhere in your lab. And what you'll want to do for each one of the different transects, you're going to calculate the various indices. The first one is the General Diversity Index. And the General Diversity Index is essentially a measurement of how the plants are spaced. So you could have something artificial like a garden where you've taken exotic species and planted them one after another, and that would be a very unhealthy, unstable ecosystem. You might also find native ecosystems where you have lots of plants that are clumped together that are the same type, members of the similar population. So in this case, what I'm taking is the total number of runs divided by the total number of plants. And as we just saw, I had 7 runs out of a grand total of 20 plants. That gives me an index value of 0.35. Now, all of these indices are graded on a scale from 0 to 1. So 0.35, not looking real good in terms of how the plants are spaced, possibly due to that big cluster of cattails that's at the very end of the data. The second index in here is the Modified Diversity Index. And the Modified Diversity Index is taking into account how many of those runs represent indigenous native species. As we saw, 5 out of the 7 runs represented indigenous species like goldenrod and sedge and cattail. So that explains why my number here is a 0.71, much higher than my General Diversity Index. The third number, the DIE, is the Exotic Diversity Index. And this really is not a measurement of how many exotic species there are, because we kind of could deduce that from the previous index here. The Exotic Diversity Index is a measurement of what kind of impact are they having on the site. So once again, you want to have a high value for this. The Exotic Diversity Index here is taken by taking the square root of the DIG times the DIM. So I'm going to take, in other words, for this case, the square root of 0.35 times 0.71. That gives me a 0.5, so not great in terms of the diversity index. It suggests that exotics are having some impact upon the site. The Species Diversity Index is just a pure measurement of how many different types of plants are present within the various runs of the ecosystem. So we found that there were 6 different species of plants: goldenrod, arrowleaf, Canadian thistle, purple loosestrife, sedge, and cattail. 6 different species amongst the 7 runs. And as you can see, that gives us a pretty high value for our Species Diversity Index of 0.86. Because we've got, for relatively few runs, we have a lot of different types of plants within those runs. But the number that matters most is the overall diversity index. And the overall diversity index here is taken as the cube root of the general times the modified times the species. This is not to be confused with 3 times the square root. That's something entirely different. You want the cube root, and that can be obtained on your calculators in a number of different ways. If you go into the math function hit the math button, you should find the 4th number down. Allows you to calculate the cube root by putting in that number. So for in this case, we took our DIO is the cube root of 0.35 times 0.71 times 0.86. That's our species, modified, and general, modified, and species indices multiplied together. What that gives us as an overall score, our DIO, is 0.6. 0.6 is not a great number. And what this suggests is that our site is not that healthy in terms of its autotroph biodiversity. And that could be due, in fact, to the fact that there are a number of exotic species that are in there, but also how they are not divided up evenly within the site. So those 2 factors are probably most significant when we assess the overall health of this ecosystem. So its score would be a 0.6. Now you would do this for each one of the transects that you did. If you did 3 transects, then you'll take the average of those 3 transects. If you only did 2, you take the average of those 2. It is also possible to get a transect calculation if you did not do a full 20 plants. Your number of total plants would just be less, and some of you did that with groups of 10.